Oh, they are very much a clinical reality. While, yes, many ADCs are in clinical trials for new targets, a significant number have already entered clinical practice. And they are providing genuinely compelling results for patients with cancers that were historically very, very difficult to treat. It's one of the fastest growing fields in oncology today.
So when we look at these clinical success stories, which target has been the most effective so far for these targeted missiles?
One of the foundational successes, the one that really proved the potential of this technology, centers around a target called HER2.
HER2, right? We hear about that a lot. Historically, very challenging. It's often overexpressed in aggressive types of breast cancer, gastric cancers. So how does the ADC mechanism play out with a HER2 positive cancer cell? Can you walk us through that sequence?
Sure. The sequence is, it's elegant, and it's perfectly designed to overcome that low expression problem we were talking about. So first, you have a monoclonal antibody that is specifically built to recognize and target the HER2 receptor.
Okay.
And this antibody is chemically linked to a highly potent cytotoxic drug.
So the whole ADC complex is in the circulation and it's just seeking out HER2 like a homing beacon.
Exactly like a homing beacon. When the ADC reaches the cancer cell surface, the antibody part binds tightly to the HER2 receptor. Now, here's the crucial step. Unlike the older drugs, the goal is not just to block the receptor from the outside. The cell senses that its receptor is bound, and through a natural process called endocytosis, it actually internalizes the entire complex. It basically invites the delivery truck inside the loading dock.
It literally opens the door for its own destruction.
Precisely. And once inside these little compartments in the cell endosomes or lysosomes, the environment changes. The pH drops, those specific enzymes we mentioned are activated, and this acidic enzymatic soup triggers the cleavage of the linker. The bond breaks.
And the bomb is released.
The highly potent toxic drug component is released directly inside the cancer cell.
Right into the cytoplasm at maximum concentration. I mean, that has to be just devastatingly effective. The cell has no defense against that kind of sudden internal attack.
It ensures the required lethal dose reaches its intended victim, while the exposure to healthy surrounding cells is minimized. And the impact on clinical outcomes, particularly for HER2 positive cancers, has been, well, it's been transformative.
Let's talk about the clinical benefit for a second. That internal release must drastically reduce the harsh systemic side effects we associate with chemo, doesn't it?
It absolutely does. I mean, think about the patient experience. Before ADCs, treating these tumors often meant using traditional chemo alongside targeted agents. So you were still suffering the total body exhaustion, the nausea of the chemo. With ADCs, we're still using potent chemotherapy, but we've managed to localize the toxicity.
So patients feel better during treatment.
We see significantly reduced systemic toxicity-less hair loss, less immune suppression, because that highly destructive agent is only activated in large amounts once it's already inside the tumor cell.
That is the ultimate trade-off, isn't it?
Right.
Delivering maximum killing power exactly where you need it, while leaving the rest of the body relatively untouched. It's a revolution in patient quality of life.
It is. For patients whose tumors express HER2 positivity, this approach has achieved just excellent therapeutic effects. It's specifically solved that historical problem. Even if the HER2 expression wasn't overwhelmingly high, just using that receptor as the delivery address for an internal explosion has dramatically improved patient outcomes.
So bringing all these takeaways together, what this all really means is that ADC drugs have found a way to deliver maximum toxicity with minimum collateral damage.